So I'm going to tell you what I think is probably one of the most interesting stories in life science, if not well appreciated. And the story begins approximately 6,000 years ago. So peyote was a medication that was used. It was the primary medication for the Native Americans of the plains. And it was primarily used as an anti-inflammatory and a pain reliever. It was used as the shaman were keen to describe as the white man uses aspirin. Now, the medicinal use of this compound was at a subperceptual basis. It was not used for its psychoactive effects. And the psychoactive effects associated with the plant were actually unwanted and incidental to its primary medicinal use. Now, flash forward 6,000 years to 2008. Our lead scientist discovers that these drugs are potently anti-inflammatory. And so Eleusis is founded in 2013 to develop serotonin 2A receptor agonists as subperceptual anti-inflammatory medicines. So our mission is to unlock the therapeutic potential of psychedelics by mitigating and managing their psychoactivity. So one of the first questions that should come up is, why am I here? Right around 2007, 2008, when this discovery was made, I was at Goldman Sachs, responsible for structured credit derivatives. And so around that time, I decided to do something new. I thought being a manager at a bank is a lot like being a psychotherapist, so I decided to enroll at NYU and to become a psychotherapist. In my studies, I came across research at Johns Hopkins which indicated that psychedelics such as psilocybin have profound potential within psychotherapy. I was intrigued, and the more I dug into it, the more I realized that the psychotherapeutic potential is just the visible part of the iceberg, and there's a massive potential beneath the surface. And that is what really kind of inspired me, because what that looked like to me is a distressed asset. Something that everyone thinks is worthless, but if developed correctly, could be transformative. And so to just give you a picture of why we think that's the case, if you look at the serotonin 2A receptor and you map it to almost every target associated with inflammaging, essentially inflammation associated with aging and chronic inflammatory disease, it seems to mediate almost all of them. Serotonin-mediated inflammation is currently a target which is not addressed because most people would assume that serotonin 2A receptor agonists can't be developed, right? It's an anti-target, and therefore no major pharma company is going to go after this particular target. However, what we've discovered is that functionally selective 2A receptor agonists have the capacity to be biased towards anti-inflammatory expression without having psychoactivity at the clinically relevant doses. And because of this, we believe that serotonin 2A receptor can be targeted with agonists, and we have evidence to support this. So we've first started in vitro, and then we've gone to translational models, and the translational model evidence is compelling. So in translational models of inflammatory bowel disease, allergic asthma, and cardiovascular disease, we've demonstrated that using serotonin 2A receptor agonists, not all of them, but some of them, that we can have clinically relevant anti-inflammatory effects at doses that would not be perceptible in humans and are certainly not perceptible in the animals based upon their stereotypical response to psychedelic drugs. So we see it in inflammatory bowel disease, we see it in allergic asthma, and we see it in cardiovascular disease. And most interestingly, in all of these conditions, serotonin, and specifically the expression of the serotonin 2A receptor, is a biomarker for the disease, right? It is upregulated in the disease state. And so the only reason why this opportunity exists is that no one was naive enough to assume that the serotonin 2A receptor was druggable, except for someone like me, a naive amateur. And because we're able to essentially question that assumption, we are going to unlock a massive therapeutic potential. Now, if these targets were not enough, we have another opportunity, which is the question is, where do we have the best safety margin, right? Because we know what the liability is. These drugs are exquisitely safe. They've been tested in thousands of studies, right? We know exactly what the toxicity is. It's a behavioral toxicity. So the best way to limit the risk of behavioral toxicity is to limit the dose and to put it into tissue where it would be confined. And that's where we get to the eye. So when we administer these drugs topically, we find something pretty remarkable, which is they get to the back of the eye via simple topical formulation, simple saline, right? No fancy formulation. Following a circumferential route, essentially transscleral, we get clinically relevant concentrations in the retina. So if we can replicate these results in humans, we've achieved in ophthalmology what is described as the Holy Grail, which is delivering a compound to the retina without any special formulation at clinically relevant doses. And if you have an anti-inflammatory compound that's also capable of exerting neuroprotective effects, then it's applicable in almost every disease state. And so these, and by the way, these results are from rabbits, which are the anatomically representative model for the human eye, right? So we're using the right model. We're demonstrating pharmacokinetically that we're getting to the target tissue at clinically relevant doses. So what is our lead compound? Our lead compound is an analog of RDOI. RDOI is a prototypical serotonin 2A receptor agonist. It's used extensively whenever scientists want to know what is the serotonin 2A receptor doing in a particular model. We have modified the scaffold to reduce psychoactivity of the compound. So essentially that modification reduces psychoactivity by roughly threefold while preserving its anti-inflammatory effect. We have composition of matter on this on the new compound, and really this is just the beginning. We have literally an extensive pipeline behind this, and the idea is that this compound is good enough for ophthalmology, but to go into the periphery, we are developing new compounds that have an even larger safety margin, where we can administer the drug orally instead of via topical eye drops. So what's our preclinical evidence? We have used RDOI and our new chemical entity in models of herpes keratitis, allergic conjunctivitis, and intermediate uveitis, essentially going from the front of the eye to the back of the eye, demonstrating that the drug is potently anti-inflammatory without having psychoactivity, right? So that's the key discovery. We've also discovered that the drug suppresses viral replication in the herpes keratitis model and prevents the animals from encephalitis. So right away, that's better than what the current standard of care is, because current standard of care in herpes keratitis is corticosteroids, despite the fact that corticosteroids actually increase viral load. So for any viral inflammation, this would be the gold standard to the extent that we demonstrate safety and efficacy in clinical trials. In allergic conjunctivitis, we've demonstrated that the drug prevents 4880 mast cell degranulation. We've demonstrated that in vitro, and we've demonstrated in the relevant rabbit model. And in intermediate uveitis, we've demonstrated that the drug suppresses inflammation at the back of the eye. But most interestingly, we've also demonstrated that the drug prevents retinal degeneration despite a profound inflammatory state induced by a tuberculosis antigen. So it's not just anti-inflammatory, but it's neuroprotective. And so that combination is incredibly captivating from our perspective, because it indicates that, hey, start with ophthalmology first, because we get absolutely low plasma levels of the drug. The drug stays in the eye. And from a pharmacokinetic perspective, we get very good tissue penetration going all the way to the retina, and we can do it in a very simple formulation. Now, if that wasn't enough, we're taking advantage of the fact that we're not the first people to think about this. Alcon spent almost 2 decades developing serotonin 2A agonists until they were acquired by Novartis. Novartis shut down the program. No one will give us a straight answer as to why they shut down the program, but we have a guess, and the guess is stigma. The reason why these drugs have not been developed to date is mostly because no one wants to develop a psychedelic drug as a basic medicine. However, there is an existing psychedelic drug already approved by the FDA for weight loss. It's called Belvic or lorcaserin, and if you take 4 times the recommended dose, you will have psychoactivity. So therefore, we have precedent. We know exactly what the safety margin has to be, and we can take advantage of the fact that Alcon already demonstrated that these drugs are effective in treating glaucoma. As effective as latanoprost, the gold standard for reducing intraocular pressure. What that indicates to us is that we've got multiple shots on goal within ophthalmology, not just with inflammation, but also interocular pressure and tear production. So in animals, we've seen an increase in tear production associated with this drug, and that's not a surprise because the leading drugs to treat dry eye disease are anti-inflammatories. And here we see the safety margin well within FDA and EMA precedent for approval. So the unmet need, diabetic retinopathy, massive unmet need, only being treated at the diabetic macular edema stage. We have the potential not only to address diabetic retinopathy at the DME stage, but to actually prevent it as a disease-modifying therapeutic. And this is just the beginning, because our next generation of drugs will be targeting peripheral inflammation. And that's where things get very interesting. NASH, asthma, all of these disease states have serotonin 2A receptor activation as a key biomarker of the disease state. I'll skip over the details. We are currently raising money to get our drug through IND and into phase 1 studies. We've had a number of venture funds that say that they're quite interested once we demonstrate human safety, and we're very confident that we're going to be able to do that. The comparison to GW Pharma is instructive. Right? This is a $4 billion market cap company that has that developed CBD, which was a stigmatized compound for an orphan indication. The indications and the applicability of what we're developing far extend beyond what GW Pharma developed. GW Pharma took a stigmatized Schedule I compound and made it into a medicine, and we are doing the same thing with psychedelics, transforming serotonin 2A receptor agonists into medicines. This is the team. Very handsome and beautiful. More importantly, tremendous expertise specifically when it comes to ophthalmology, when it comes to drug development in the serotonin 2A receptor space. We have 2 sponsored research teams at LSU. We are a UK-based company with US and UK operations. This is where we're covered in the media. The stat news piece is probably the best starting point. We've also conducted clinical trials with LSD and Alzheimer's, and I'll save that for another day. Thank you.